all for coming today, and it's my great pleasure to introduce Dr. Yanlong Song, who, as some of you may know, is one of the first recipients of our See Progress pilot funding. And so we are thrilled to welcome him today and to learn more about his work. Dr. Song is currently a postdoc research fellow at Cook Children's Health System and the University of Texas at Arlington. His research interests center on the learning and control of manual movements and the related neural processing alterations in patients with movement disorders. His research at Cook Children's focuses on assessing cortical sensorimotor functions in children with cerebral palsy, with MEG, high density EEG, and TMS. So lots of interesting information for us about those. Before joining Cook Children's, he obtained his doctorate degree at Iowa State University in kinesiology, focused on human motor learning and control. And then his first postdoctoral training was at the University of Virginia, where he studied TMS brain stimulation. So we are thrilled to welcome you here today, Dr. Song. Thank you so much for joining us, and I will let you take it away.
Okay. Thank you, Dr. Nari. So let me share my screen. Okay. So I think you can see my screen and see my pointer, right?
Yes.
Okay, thank you. So today I will talk about aberrant sensorimotor processing in children with hemiplegic cerebral palsy. I will discuss some findings from MEG and the EEG brain imaging. So the motor project of this presentation is funded by See Progress. So my mentor and supervisor, Dr. Christos Papadakis, is also here. So, okay, now let's start. So I want to start to talk about the human hand. We all know that the hand is one of the most important parts of the body. It can perform so many movements with delicacy and precision. Many movements like reaching and grasping, holding, typing, writing, all these movements, they play very critical roles in our daily independent living. Impairments or loss of any of these functions will usually severely impact the quality of life. So when I think about how can we do so many manual movements with delicacy and precision, I think one critical reason is that we have very advanced central control of head action. Specifically, we have very fine cortical representations in the somatosensory cortex and the motor cortex. So this figure is the famous Penfield homunculus. You can see here around in the somatosensory cortex and the motor cortex, the hand takes big part of these sensorimotor cortex areas, and the fingers, they have a very orderly representation. And the second aspect is that we have a very distributed motor control network. Most parts of the cortex and the basal ganglia and the cerebellum, they function as an integrated dynamic neural network in the controlling of head actions. This dynamic neural network, they are performing advanced parallel computations on the processing of sensory feedback and the prediction of movement-related sensory consequences. So disruption of any one node of this network will usually cause the disruption of the network performance. For instance, the damage to sensorimotor cortex can usually cause hemiplegic, disruption of the control of fine movements. This kind of phenomena often occur in children with cerebral palsy. Cerebral palsy is the most common physical disability in childhood. It affects about 1.5% to 4% of US live births. Among children with cerebral palsy, 30% to 40% will develop hemiplegic. So we term this subgroup of children as hemiplegic cerebral palsy, HCP in short. The etiology of HCP is really complex. But like early brain injury from like damage to paraventricular white matter or middle cerebral artery blockade can usually cause cortical damage. Cortical damage near or in sensorimotor cortex can usually cause impairments in the contralateral upper limb and the lower limb functions. And in most cases, the upper limb impairments is most severe. This figure shows like common thing, manual impairments in children with HCP. You can see some children may show excessive thumb adduction. Some children may show excessive finger flexion. Some may have difficulty to open fingers. Some have difficulty to individually control finger. Some children also have very limited wrist extension. All these impairments will limit children with HCP to perform daily activities like grasping, like writing, these kind of movements. And the recovery of many functions is often very difficult. So in the following, let me to minimize this, okay. So in the following, I will firstly talk a little bit about MEG and EEG, and then I will talk about cortical motor processing and the cortical somatic processing. MEG and EEG provides direct measures of neural activity of the cortex. So where do the signals of MEG and EEG come from? So a well-accepted view is that MEG and the EEG signals, they are coming from postsynaptic primary electric currents. Because of the morphology of neocortex pyramidal neurons, these postsynaptic primary currents can be modeled as a small dipole, as shown here by this red arrow. It has orientation normal to the cortex mantle. So these postsynaptic primary currents will generate volume currents. The volume currents will propagate through different tissues, include cortex, spinal fluid, skull, and scalp. And if we place 2 electrodes on scalp, the signals collected is EEG signal. And meanwhile, the primary currents and the volume currents can generate very weak magnetic fields. If we place a very sensitive magnetic sensor near scalp, the signals collected are MEG signals. Both MEG and EEG, they have their pros and cons. The first aspect is they have to deal with signal decay. EEG measures volume currents. I have said before, the volume currents propagate through different tissues. And these tissues will absorb a portion of EEG signal. And more importantly, these tissues have different conductivity. This will cause difficulty for later if we want to localize the source origins of EEG signals. Differently, the magnetic field can pass through all the tissues without almost no loss. However, the detectability of a magnetic field is greatly reduced when the distance between the sensor and the field increases. So that's why we rarely see findings on MEG on subcortical regions like basal ganglia, this kind. And the second aspect is signal sensitivity. So if the brain is modeled as a sphere, so the neuronal sources can be grouped into 2 types. The first type is the radial component. They are mostly exist in the gyro locations. And the second is tangential component. They mostly exist in source regions. EEG measures volume currents, so EEG are sensitive to both tangential and radial components. However, MAG is only sensitive to tangential components because the magnetic field from radial components will cancel out. MAG and the EEG signals can be analyzed at both signal level or source level. To localize the source origins of MAG and EEG signals, we have to perform the following three steps. The first one is to register MAG or EEG signal to structure MRI. This is currently done by using three-dimensional digitization. So we will use a 3D digitizer to digitize landmarks like lesion, LPA, RPA, and several hundreds like actual scalp points in MAG, and also EEG electrode locations. And then by use MRI landmarks, we can register MAG or EEG signal to MRI and form a correlation file. The second problem is the forward problem. So this problem is given that we know the location of a source origin. How will this source origin generate signal distribution on scalp? So to solve this problem, we have to have a head model. A precise head model is especially important for EEG. So this is the example of a BEM model. It consists of 3 layers: cortex, skull, and scalp. And the conductivity of these layers is estimated from structure MRI. So by combining this head model and the co-registration, the forward problem can be solved. And the third problem is the inverse problem. So this problem is given we have the signal distribution from MAG or EEG. How do we know where are these sources generated? So this is the ill-posed problem because the possible numbers of source origins are usually more than the number of MAG sensors or EEG electrodes. So from the perspective of mathematics, this problem doesn't have a unique solution. So to solve this problem, we have to place some prior constraints. A commonly used constraint now is that we constrain all the sources to cortical green matter. So by combining the solution for the forward problem and these constraints, we can localize the source origins for EEG or MAG signal. At Cook Children's, we are using this Meijin NeuroMag system. It has 306 sensors. All these sensors, they have to function in a very super low temperature environment. So that's why this machine has this big part. It's used to store liquid helium to create a super low temperature environment for the sensor to function. EEG we are using is EEG geodesic system. We have caps with 256 electrodes or 128 electrodes. These caps are compatible with MAG systems, so they have very high tolerance to impedance. Considering potential difference to the source orientations, so we are using simultaneous MAG and EEG recording whenever it's possible in our studies. So let me translate to the motor deficits in children with HCP. So many behavior studies have reported motor impairments in children with HCP, including like high muscle tone, involuntary contraction, behavior movements, difficulty to individually control finger movements, and also like they have poor fine motor skills. All we have studied is the precision grip. So in children with HCP, they have difficulty to exert proper amount of precision grip force in the affected hand. Children with HCP, they also have difficulty on bimanual coordination. Only a few MAG or EEG study examined neurophysiological alterations in these children and reported potential like changes in gamma during motor execution and the beta after movement cessation. So in this study, I aimed to further assess potential alterations of cortical motor processing in children with hemiplegic cerebral palsy. So this figure is about experimental setting up. You can see here, the child is sitting here and undergoing MAG and EEG recording. And in front of him, there's the screen. And this figure is a behavior paradox. We're using a go, no-go behavior task. So you see, after the fixation cross, a visitor go cue, either a cartoon dog or a cartoon cat will be displayed. When the cartoon dog is displayed, the child is instructed to tap his right index. And when a cartoon cat is displayed, the child is instructed to tap left index. In 25% of these trials that randomly selected, immediately after the presence of dog or cat, at about 150 milliseconds to 250 milliseconds later, a red light bulb will be displayed. This red light bulb is to indicate the child should withhold his tapping or stopping his tapping. So by using this go and no-go behavior task, I wanted to tangle the motor planning process, because this task, to correctly perform this task, the child has to make correct decisions. Otherwise, he will make errors. So this is an attention demanding task. So we only test the children aged 10 years and older. And this project is funded by Cerebral. So before I talk about these slides, I give you a little background. Many studies in healthy adults, they reported manual movements modulation of cortical oscillations, mostly in alpha and beta frequency bands. That's before movement execution, alpha and beta oscillation power will decline relative to baseline phase. This is termed as event-related desynchronization. So here's this figure. This is a time frequency map. The y-axis is frequency and x-axis is time. So the value for each time and frequency pair represents the power change relative to baseline window. The baseline window here I used is from -1 to -0.5 seconds. So for this chart, you can see this time frequency map is the average of all 306 MAG sensors. When the trials all aligned to the Q onset, and in this condition, it's a cat, and the child is instructed to tap the left index. We can see here, after about 200 milliseconds after the presentation of the Q, the alpha is depicted by this green window, and the beta, here, this orange window. Both alpha and the beta power declined relative to baseline phase. So this is ERD, event-related desynchronization phenomenon, you can see here. Similarly, when a cartoon dog is displayed and the child is instructed to tap right index, we can also see here alpha and beta ERD in this condition on the MAG sensor level. So after several steps of processing, I can localize possible source origins of alpha and beta ERD. So this figure is the source localization of alpha ERD when the child is instructed to tap left index, you can see here. Most alpha ERD is generated in occipital region, and the posterior parietal region, and maybe also the sensorimotor regions. This is the same for both conditions. And for beta ERD, we can see, besides occipital region, we see very clear generation of beta ERD in sensorimotor region. And more importantly, we can see here, there may be a contralateral dominance for the generation in the sensorimotor cortex of beta ERD. You can see this is when the child is instructed to tap left index, so the right sensorimotor area generated a stronger beta ERD. And when the child was instructed to tap right index, the left sensorimotor cortex generated a stronger beta ERD. So after moment cessation, and when all the trials are aligned to the tapping onset, you can see here, alpha and beta power increase relative to the baseline window. So this kind of phenomenon is termed event-related synchronization. And here, this is a child tapped left index, and this is the child tapped right index. We can see both evident alpha and beta ERS in both conditions. And we can also localize the source origins of alpha and beta ERS. But a point I have to mention is that we don't know clear about the functional roles and the source origins of alpha ERS. There is no well-accepted view on this, so we can ignore alpha ERS. But as to beta ERS, okay, so you can see here, when the child tapped left index, the right sensorimotor cortex generated much stronger beta ERS. And when the child tapped the right index, the left sensorimotor area generated much stronger beta ERS. So this kind of beta ERS is thought to reflect the reset of sensorimotor cortex after moment cessation. This is an accepted view. So this is the data in a healthy, typically developed children. So now let's move to the data for children with HCP. So you can see here, similar as the typically developing children. So on the max sensor level, so when the trials aligned to Q onset and the child is instructed to tap the left index, we can see alpha and beta ERD. And similarly, when the child is instructed to tap right index, we can see alpha and beta ERD. However, we can see differences for the source localizations for alpha ERD and beta ERD. If you still remember that for alpha ERD and beta ERD, in the TD child, occipital region, posterior parietal region, and the sensorimotor cortex, these areas generated very strong alpha and beta ERD. However, in this child with HCP, you can see the child has a big lesion in the left hemisphere. The child only show alpha and beta ERD in occipital region, you can see here. But this child doesn't show the generation of alpha ERD and beta ERD in posterior parietal cortex and the sensorimotor cortex. And we can also not see any contralateral dominance pattern in the generation of beta ERD. So this potentially indicating the alteration of motor planning process, the disruption of motor planning process in this child. And when the trials are aligned to the tapping onset, you can see after the tapping cessation, similar timing. So we can see alpha and beta ERS in both conditions when the child tapped the left index or tapped the right index. However, let's see the source localization for the beta ERS. So you can see when the child tapped left index, we can see the right sensorimotor cortex may generate some like beta ERS. And similarly here, this is when the child taps the right index, the left sensorimotor cortex also generates some beta ERS. So here, this is the average of 5 typically developing children and the 5 children with HCP. So you can see on MAC sensor, this is the average of all the MAC sensors. So compared to typical developed children, although we can see alpha and beta ERD and alpha and beta ERS in children with HCP, however, the magnitude of alpha and beta ERD and alpha and beta ERS are clearly like weaker than that in typical developed children. So here, this is the results of group average of source localization for alpha and beta ERD. So we projected all the source localizations of all the children to a template, MRI template. So you can see here, this is source localization for alpha ERD. When typically developed children were instructed to tap left index, you can see most alpha ERD is generated in sensorimotor, in occipital region, posterior parietal region, and also sensorimotor region. This is also the same when the child was instructed to tap right index. And similarly, for beta ERD, we can see similar pattern. And moreover, as to the sensorimotor cortex, we can see a very clear pattern of contralateral dominance. We can see when the child is instructed to tap left index, the right sensorimotor cortex generated stronger alpha and beta ERD. And when child is instructed to tap right index, the left sensorimotor cortex generated stronger alpha and beta ERD. So that to see alpha and beta ERD likely reflects multiplanning related process and the activation of visual motor transformation pathway. This pathway including the occipital region, posterior parietal region, and the sensorimotor region. Okay, so in sharp different from typical developed children, children with HCP you see here, only the occipital region show clear generation of alpha ERD when the children were instructed to tap left or right index. However, we couldn't see clear generation of alpha or beta ERD in parietal region and also sensorimotor region. So this is a clear evidence for the disruption of visual motor transformation pathway and the motor planning process in children with HCP. Here, this is the group average results for the source localization of alpha and beta ERS. Okay, so let's focus on the beta ERS. So for the typical developed children, when the child tapped the left index, we can see the right sensorimotor cortex generating much strong beta ERS. And when the typical developed children were instructed to tap right index, the left sensorimotor cortex generated very stronger beta ERS. So there is a very clear contralateral dominance pattern in typical children. For children with HCP, although we can see when they tapped left index here, right sensorimotor cortex generates beta ERS. Okay, when they tapped right index, so the left sensorimotor cortex generates beta ERS. However, the amplitude for beta ERS is not so strong as that in TD children. So maybe they also show some kind of disruption of motor resetting, the resetting of sensorimotor cortex in children with HCP. Sensory feedback is very critical in the fine control of manual functions. So is it possible for us to get sensory feedback related signals in finger tapping? Yes, it is. So this figure is the average finger tapping evoked response across all the MAC sensors. So here you see all trials are aligned to the tapping onset. And after about 60 milliseconds, there is a very clear and sharp peak and trough, and we call this is a component. This component is termed motor evoked field. So for this TD child, when he tapped his left index, we can see this motor evoked field is localized to right sensorimotor cortex, and with a maximal actually localized to post-central sulcus to the contralateral somatosensory cortex. So this motor evoked field actually mostly reflects somatosensory feedback. And similarly, we can see very clear and sharp motor evoked field in 3 other children when they tapped their index. I wanted to pay attention to the magnitude of this motor evoked field in these TD children. However, in children with HCP, motor evoked field is greatly reduced even when they tap the less affected head. We can see, although there's a like smaller component, but in these two children, there's no very clear motor evoked field when they tapped their less affected hand. And more importantly, when children with HCP tapped their affected hand, motor evoked field, we cannot see any motor evoked field in this kind of tapping. This is the indication of the disruption of somatosensory feedback in these children. Somatosensory feedback is very critical for the fine control manual functions. This is especially when we are familiar with the movement and we don't rely so much on vision. Many studies have examined somatosensory deficits in children with HCP and reported behavioral changes like diminished tactile discrimination, diminished joint position sense, and kinesthesia. MAG or EEG studies reported neurophysiological changes like including abnormal contralateral primary somatosensory S1 evoked response, abnormal somatotopy of finger representations in somatosensory cortex, and the somatosensory oscillations, and also abnormal somatosensory functional activity. And more importantly, somatosensory deficits are correlated with motor deficits. So in this study, we are using simultaneous MAG and high-density EEG recording to further assess potential alterations of cortical somatosensory processing in children with HCP. Here, this child sits here and undergoing simultaneous MAG and high-density EEG recording. And during this recording, we use pneumatic stimulator to randomly stimulate thumb, middle finger, and pinky of both hands. Each digit is stimulated about 400 times. So here I present you somatosensory evoked fields and potentials in a TD child. You can see this is the somatosensory evoked fields across all MAG sensors when the right middle finger is stimulated in this child. You can see here, there are 3 very evident components. The first component occurs around 50 milliseconds, and the second component occurs at 100 milliseconds, and then the third component occurs at about 150 milliseconds. And here, this is the source localization for the first component. You can see the first component mostly localized to left primary somatosensory cortex, although we also can see a little activation at the secondary somatosensory cortex. And at the second component, you can see the maximum activation shift from left S1 to left S2 area. This is an indication of the somatosensory transmission. And at the third component, although the maximal activation is still in left S2 area, and we can see activations in right S1 and S2 areas. So by localizing these three components, we can roughly judge the transmission of somatosensory information following a pattern from contralateral S1 to contralateral S2 and then to ipsilateral hemisphere. The pattern for evoked potentials in EEG is more complicated. For this chart, you see, EEG has like 7 components. So the first component occurs around 50 milliseconds too, and it localized to left S1 area. And the second component, the maximal is still in left S1, but we can also see some activation in left S2. And at the third component, we can see activations in the midline areas and the ipsilateral hemisphere. Here, this is the results from a child with HCP. So this child, you can see, he has a very big lesion in the right hemisphere. This is the MAC evoked response when the right middle finger is stimulated. We can see a very sharp first component around 50 milliseconds. And this component is localized to left S1. And then at the second component, around 100 milliseconds, we see the information being transmitted to midline area. And the third component, the information transmitted to left S2 area. If you still remember the localization for the TT child, at the third component, around 150 milliseconds, the TT child show activations in the ipsilateral hemisphere, but this child didn't show this kind of activation, likely because of its large injury in the right hemisphere. And when the left thumb is stimulated, we see 2 very evident components. And the first component is localized, very different from typical TD child, localized to the right S2. Although so, we can see very strong activation at the edge of the lesion. This is likely maybe from residual S1 activation. And I think this kind of residual S1 activation is very critical for the manual functions. This photo shows how this child is picking up a paperclip. You can see his position of thumb and the index is very good, and he can quickly pick up this paperclip. At the second component, the activation is still at the right S2 area. This is another child with HCP. This child has a small lesion, but near sensorimotor cortex in the left hemisphere. So when the left middle finger is stimulated, you can see here, the first component occurs around 50 milliseconds, but it's maximal localized to not to right S1, but to right S2 area. So this is a sharp difference from that TD child. And although this is the less affected hemisphere, but you can see at the second component, the activation pattern is completely different from that TD child. And when the affected hand, the right middle finger is stimulated, we didn't see a clear component around 50 milliseconds. So this first component may be missed or delayed. And around 100 milliseconds, there is a component, and this component is most localized to left S2 area. And we barely see any activation around the left S1 area. That to say, the loss of activation in or near contralateral S1 is very critical. And this child mental function is actually not very good. This further shows how he is picking up a paperclip, and you can see how he positions his index and thumb. And actually, he traveled several minutes and he couldn't pick up this paperclip. This is another child with HCP. He has large lesion in the left hemisphere. Let's focus on the stimulation of the affected hand. You see, this is the stimulation of right middle finger. You can see pneumatic stimulation actually doesn't evoke any clear component in this kind of condition. And around 15 milliseconds, this is the socialization. Most activation actually occurs in frontal cortex. We couldn't see any evident activation in either left S1 or left S2 areas. And this child has very strong difficulty to open fingers, if you can see from this figure. So by randomly stimulate thumb, middle finger, and the pinky in both hands, we can reliably localize S1 and S2 areas in both hemispheres. By using these localizations, we can do further processing like time frequency analysis, functional connectivity analysis. Here, I give you an example of the group average of contralateral S1 activation in 9 TD children and 9 children with HCP. This is the average from MAC signals. You can see, compared to typical Didactic children, children with HCP show much weaker contralateral S1 activation. This is also supported by time frequency analysis. So this is the difference between time frequency analysis between TD children and children with HCP by using signals extracted from contralateral S1. You can see here, around 50 milliseconds to 100 milliseconds near beta frequency band. Children with HCP show much weaker activation than TD children. However, this is not so evident in EEG. So let me summarize up. So from these findings, we can see likely disruption of visual motor transformation network in children with HCP. These children, they show reduced alpha and beta ERD in posterior parietal cortex and the sensorimotor area. They also show reduced beta ERS in sensorimotor cortex. And they lack contralateral dominance in sensorimotor area in the generation of alpha and beta ERD and ERS. And we also see evidence for potential alteration of somatosensory processing. Specifically, children with SCP show reduced somatosensory feedback accompanying finger tapping, and they show weak early contralateral S1 activation, but strong early contralateral S2 activation. So I want to finish my presentation by mentioning individual variation and individual variability. I didn't list it here. Every child with cerebral palsy is unique and special. They have different locations of lesion, different time of lesion, different functional levels. So I think this kind of brain imaging studies with MAG or EEG will provide us with critical information to understand their brain and the cortical information processing. This may be helpful for further like management and functional recovery. At last, I want to thank my team for supporting me in my research. I especially want to thank my current supervisor and mentor, Dr. Christos Papadakis. He taught me a lot of things from MAG, data analysis, somatosensory stimulation, and thank you very much, Christos. I also want to thank Dr. Marks and Dr. Kurt for introducing their patient to us. Lastly, I want to thank CProgress and the ISCPDM for funding the motor project in my study. Thank you all for your attention.
Thank you so much, Dr. Sung. That was wonderful. So my job